Hi everyone and welcome to AP Help 101. So actually I'm going to just start breaking down some of the subjects that could possibly be on the test. So this week I'm going to start with biochemistry. So atoms. We're going to talk about atoms. There's a lot to talk about actually. There are three parts to an atom. There's a proton, a neutron, and an electron. And a proton has a positive charge, a neutron has a neutral charge or no charge, and an electron has a negative charge. Ground state. Ground state is basically when an atom is at its lowest energy level. It has the least amount of energy at this time. An excited state is when an atom is at its highest energy level. So as the atom is gaining more and more energy, it's going to go to a higher excited state. Isotopes. Isotopes are basically elements that have the same number of protons but different number of neutrons. So for example, we have carbon-12 and carbon-14. They both have 6 protons, which makes them carbon, but they have different numbers of neutrons. Carbon-12 has 6, carbon-14 has 8. So basically the atomic mass changes, but it's still carbon. Now there are two types of bonds. An ionic bond is when atoms give up electrons. So basically they don't share them, but they completely give them up. So one becomes a cation, which is a positive, or an anion, which is negative. And a covalent bond is when atoms share electrons, which results in a molecule actually. So there are two types of covalent bonds, nonpolar and polar covalent. A nonpolar bond is when electrons are shared equally, and this usually happens in two of the same element bonding. So, for example, H2 or O2. A polar covalent bond is when electrons are not shared equally, and this usually happens when two different elements bond, such as CO2 or H2O. One of the elements usually gets more of the electrons than the other element does. So in carbon dioxide, the oxygens are going to get more of the electrons than the carbons, and that's actually called electronegativity. And so elements like oxygen and fluorine, they have a higher electronegativity, meaning they're more likely to take away electrons. Now hydrophobic means water fearing. So for example, oil, it doesn't mix well with water. And hydrophilic means water-loving, such as salt, which dissolves very easily with water. So now we're going to talk about properties of water. And there are 5 key concepts to remember. 1. Water is a universal solvent, so a lot of things dissolve in water, basically. I mean, obviously things such as oil doesn't dissolve, and hydrophobic things don't dissolve in water, but everything else does. Now, water has a strong cohesion tension. So what that basically means is that water molecules attract each other, which allows water to defy gravity. And you can see that it's called capillary action. And you can see that in plants when the water goes up from the roots to the leaves of the tree, or the flower, or any type of plant, but water is defying gravity basically. Water also has a high specific heat. So lots of heat is required to drastically change the temperature of water, so this actually allows for a more stable environment. And actually, the marine biome is probably the most stable biome on this planet because it takes a lot of heat for the water to drastically change temperature. Water also has a high heat of vaporization. What that means is that when water evaporates, the surface becomes cooler. So when you sweat and that sweat is evaporating, you automatically become cooler. And finally, ice is less dense than water. And so this is how ice can float on water. When the water freezes, the particles actually space out a lot more, therefore making ice less dense than water, allowing it to float. Carbohydrates. Carbohydrates are made up of 3 elements: hydrogen, oxygen, and carbon. A monosaccharide is just a monomer of carbohydrate, for example, glucose. A disaccharide is two monosaccharides put together, for example, lactose. And a polysaccharide is polymers of the carbohydrate. So there are many monosaccharides. So for example, cellulose is an example. And this is just a picture of the monosaccharide, the disaccharide, and the polysaccharide. So you can see mono is one, di is two, poly is many. It's a chain. Lipids. All lipids are hydrophobic, so they do not dissolve in water at all. So for example, we have fats and oils. Lipids have a couple of different functions. Energy storage, they make up membranes, and they help out with hormones. Usually that's actually steroids. They have 3 fatty acids and a glycerol. That's what most lipids are made up of. But steroids are actually an exception. These are made up of 4 fused rings put together. For example, cholesterol. Now there are 2 types of fatty acids. There's a saturated fatty acid, which is only a single bond between carbons. So every single bond between carbons is single. There's no double, no triple bond at all. These are solid at room temperature, and they're usually found in animals. An unsaturated fatty acid has at least one double or triple bond between the carbons. So even if there's just one, it automatically becomes an unsaturated fatty acid. And this actually forms a bend in the molecule, and I'll actually show you that. So this is cholesterol. This is a steroid. You can see the 4 fused rings. And this is a saturated versus unsaturated fatty acid. So the saturated, you can see, is just a straight line of single carbon bonds, but you see the double bond for the unsaturated fatty acid right there, causing a bend. Phospholipids. These have a hydrophilic head and a hydrophobic tail. And they build themselves in a double layer structure called a bilayer. So the tails come together and the heads are sticking out because these phospholipids are usually found in water. They're your membranes. Your membranes are made up of phospholipids. So you can see the heads on the outside and the tails on the inside because the heads like water and the tails don't like water. Proteins. Proteins also have a couple of different functions. They're useful in growth and repair, signaling, hormone regulation, enzymes, and in movement. A monomer of protein is an amino acid. It's made up of a carboxyl group, an amino group, and an R group. And the carboxyl group and the amino group don't change, but the R group does. The polymer is a polypeptide that's just a long chain of amino acids. Now there are 4 levels of structure here. We have a primary, which is a sequence of amino acids. Secondary, which is the alpha helix and the beta pleats. Tertiary, which is a 3D shape, and it determines specificity, and quaternary, which is basically more than one polypeptide. And a lot of proteins actually don't make it to the quaternary stage. A lot of them actually just stop at tertiary. So quaternary is not in all of them, but it's in some. And as you can see, these are the 4 levels of structure. You can see the amino acid and the alpha helix and the beta pleat. And you can see the 3D shape actually with all the bending. And then finally the quaternary for some, which is many polypeptides put together. Nucleic acids. There are 2 types of nucleic acids, ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. The monomer of nucleic acid is a nucleotide, which is made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base. And the nitrogenous bases are adenine, guanine, thymine, and cytosine. Now, a polymer is, of course, the nucleic acids, which is just the nucleotides. And this is an example of what a nucleotide looks like. You can see the phosphate group on the very left, then you see the 5-carbon sugar, and finally the nitrogenous base. So we're going to talk about energy here. The first law of thermodynamics, which is energy cannot be created or destroyed. So it can, change form, but you cannot all of a sudden create energy, and you can't all of a sudden destroy it completely. It's still there. The second law of thermodynamics is during energy conversions, the universe becomes more disordered. And so those are the two laws of thermodynamics. Just keep that in the back of your mind. It might be a question on the test. An exergonic reaction is where energy is released, and an endergonic reaction is where energy is created. And so those are important to remember, as well as exergonic reactions power endergonic ones. Enzymes. So an enzyme has an active site where a substrate fits in. So really, that's where the action takes place, which is in the active site. Each enzyme is specific for a certain substrate, so not just any substrate can fit in an enzyme, it has to be that specific one. And the enzymes have a specific temperature they function at. If it's too high or too low, then the enzyme denatures. And that also works for something such as pH. If the pH is too high or too low, the enzyme is going to denature. And that's the reason why our body needs to stay at a certain temperature, is because the enzymes in our body won't be able to function. They won't be able to do their job if our body temperature goes too high or too low. So this is just a very basic picture of an enzyme. You see the substrate and the active site. The substrate and the enzyme, you can see, go in together like two pieces of a puzzle. And the active site is where they come together, and that's where they catalyze the reaction. Enzyme inhibitors. We have two types of inhibitors. The first one is a non-competitive inhibitor, and it attaches to the allosteric site. And I didn't point that out in the last picture, but I have another picture that'll show the difference between the two inhibitors, actually. So when it attaches to the allosteric site, it changes the shape of the active site so that the substrate cannot come in, and it won't be able to catalyze its reaction. And with the shape change, the substrate won't fit in like two pieces of the puzzle. A competitive inhibitor mimics the substrate, and it attaches to the active site and prevents the substrate from coming in. And it doesn't attach and stay there, it attaches and comes out, attaches and comes out. But it does that so many times that it's hard for the substrate to come in, so it'll catalyze the reaction. And therefore, that's how it inhibits the process. So this is just what the different types of inhibition looks like. So you can see the normal binding first, where the substrate fits perfectly in the active site. Then you see the competitive inhibition, where there's the inhibitor right there preventing the substrate to come into the active site to catalyze the reaction. And finally, you see the non-competitive inhibitor, which you can see the allosteric site on the other side of the enzyme, on the other side of the active site. It attaches there, and then it changes the shape, and now the substrate cannot fit in to the active site, and it cannot catalyze the reaction. So I hope you enjoyed this video, learned a lot about biochemistry, just refresh your memory. Leave comments below, suggestions as to what to do next, and I'll see you guys next week.